All right, everyone. Novel psychedelic molecules or new chemical entities are a hot topic in the psychedelic zeitgeist right now. You hear lots of talk and hype about them, but we never really get into how they're created or the challenges that are involved in creating them. Oftentimes, when you hear from these companies that are working on new chemical entities, it's sort of like you get a bunch of money, you throw it at some scientists, and then all of a sudden you have novel psychedelic molecules. And today's guest, Sam Banister, is going to sort of demystify the process of novel psychedelic molecules and tell us how he's applying some of his skills at generating new molecules to his new company, Psylo. Sam is a medicinal chemist who is He's done lots of work on novel cannabinoids in his academic research, including at his postdoc at Stanford. And now he's co-founder of Psylo, and he's joining us all the way from Sydney, Australia. Is that right, Sam?
Yeah, that's right. Yeah, just
Yeah.
at home here, Brom, instead of in our beautiful new offices at UNSW for Psylo.
That's awesome, man. And even his home is psychedelic themed. If you're watching the video, you can see he's got LSD blotter art behind him. Undipped, he told me before, but I don't know if that's true or not. We'll assume it is. One thing, Sam, that I just thought of now that I don't know if this has been covered before, but like, what is sort of the psychedelic scene like in Australia? I'm a very US biased person, so I kind of know what's going on around me, but like, is there as much hype there as there is, in the other parts of the world?
Yeah, it's a really good question, Brom. It's obviously we're sort of somewhat geographically isolated here down in the Pacific, a little bit further from everyone else. There's a huge amount of interest at the moment in psychedelics in Australia. So the federal government last year announced a research fund to the tune of 15 million Australian dollars to fund a number of psychedelic clinical trials here. You've got a number of cannabis companies setting up their own sort of subsidiaries or related entities to do psychedelics work now, or taking in licensing compounds from universities in that space. Quite a few nonprofits. There's the Psyche Institute was just set up with a about $30 million donation. $30 million of funding, from an undisclosed North American biotech to do some clinical trials work here. So yeah, I'd say there's a huge amount going on,
Wow.
and obviously Psylo's heavily focused on the sort of NCE space and next gen psychedelics.
Right. And that's so obviously there's a lot of interest from the research side, but in terms of just the, cultural side, I mean, what's the status of is cannabis legal in Australia? Or
Yeah,
like,
yeah. So
I
we're a
don't, I
little
don't
bit.
even know.
If you look at the World Drug Survey data, which is all sort of self-reported data from around the world, apparently we love drugs more than average. Apparently, we have one of the highest rates of cannabis use,
Okay.
some of the highest prices for cocaine. So we definitely love and use drugs. A sometimes problematic relationship with alcohol culturally, I would say.
Gotcha.
So drugs are definitely popular here. Cannabis is not recreationally legal. ACT, I believe, which is one of the territories here, like a state, has allowed people to grow cannabis for quite some time. We have an ever-growing medicinal market, so it's now through a special access scheme, which is for unregistered medicines. It's now possible to get a number of cannabis products relatively easily for a number of indications. But I'd say we're still sort of several steps behind a lot of what's happening in the US at the state level and Canada and elsewhere.
That's interesting. Okay, that's cool. Yeah, it's always interesting to learn about, like the drug situation and different parts of the world. That's very cool. Okay, so let's talk novel chemical entities, new chemical entities. Your whole career has kind of been around this idea, right, of generating novel compounds. Is that sort of fair to say?
Yeah. I started out as a medicinal chemist a number of years ago. Got my PhD here looking at a class of compounds that are cage-like amines, polycarbocyclic amines, for treating things like cocaine addiction via the sigma-1 receptor. And I've stayed sort of broadly in drug development since then. And a lot of drug development is, molecular design and optimization, thinking about how a small molecule interacts with a particular protein target and ideally developing drugs to treat patients.
Right. And you did some work with the cannabinoids, right? And I think some of your
Yeah.
synthetic cannabinoids ended up getting pretty popular for maybe without your intent. Is that
Yeah.
correct?
Yeah, that's right. So I wouldn't be the only case. John Huffman is the person whose compounds were first brought to market, without his approval as new psychoactive substances. I fell into that space sort of accidentally because one of the structural components of some of the compounds from my PhD ended up being detected in a new cannabinoid. At that time, it wasn't clear it was a cannabinoid; it was a completely new. We did a little bit of structure activity work, which is pretty typical for medicinal chemistry. You change one feature at a time and see how that affects activity at cannabinoid receptors in this case. And we gave those compounds a coding system like any chemist does. So I just used my initials SDB 001 through 12. And yeah, a couple of those turned up in Europe from Hong Kong, so presumably manufactured in China, shipped out of Hong Kong to a number of
That's
countries in Europe to be sold on the sort of kilogram scale, which was rather concerning email to receive from a number of Interpol and a number of heads of police in Europe asking for more information around this compound that I'd sort of designed and studied.
That's so crazy. Yeah. And I would say, I don't want to like repeat all the stuff that you said on the Hamilton Morris podcast, but I'd say if you want to learn more about that, Sam did an awesome podcast with Hamilton Morris that you should check out on Hamilton's Patreon. Definitely worth a listen. So one last question about synthetic cannabinoids. Do synthetic cannabinoids have a synthetic entourage effect?
These are unregulated products, so you're probably
Okay.
at any one time. I mean, if you look at data from Europe, where people do a lot of analysis of these substances, often the commercial products that are sold in these bright, shiny foil packaging, often they contain more than one compound. So who knows what sort of
That's true.
entourage you're getting
There
from.
may be more of an entourage effect in the synthetic stuff than in the natural
I
stuff.
think so. Yeah, I think so.
Okay. Yeah, for those who are kind of out of the loop there, Sam is a very vocal critic of the entourage effect on Twitter, as he should be. Very again another topic that they cover at length in the Hamilton podcast that I recommend you check out. So I posted something on Twitter earlier today about in 10 years from now, what's going to be more popular, psilocybin and LSD and MDMA, or is it going to be these next generation things? And you see a lot of people saying, oh, well, it's going to be psilocybin and LSD forever. And then you said, well, maybe 10 years from now, those will still be popular, but 20 years from now, the new stuff is going to be dominant. Maybe a good place to start would just be, what are the motivations for creating novel psychedelic compounds? Like, why do we need to create something besides psilocybin or LSD? Those are so great. Like, what's the point in improving them?
Yeah, I think there's a number of different arguments to be made here. And I guess people like Alexander Shulgin and even Albert Hoffman have focused on the design of new psychedelics purely out of academic curiosity. What is the relationship between structure of a molecule and psychedelic activity? Perfectly valid scientific question, and obviously a huge amount of chemical space to be explored there. From a more sort of clinical lens, these drugs won't be suitable for everyone, right? And this is why you have companies like Psylo and a number of other companies looking at NCEs in this space. So it's very rare that a natural product, as it's found in nature, is perfectly optimized for human use. And that's because these molecules are not made, we're very anthropocentric. We think about this mushroom growing somewhere to produce this magical substance for our specific use. Obviously, that's not the case. It's for reasons that are unclear to us, possibly for defense, possibly to encourage spreading of the mushroom. They produce these substances that are not optimized for clinical use. So one of the immediate issues that groups like Compass and others will be aware of is that there's a limit to how you can scale psilocybin assisted psychotherapy if the duration of action is sort of 6 to 8 hours and you have to have, two people supervising at all times. It's, that there are serious barriers there to scaling that operation as a medical product. So shorter acting tryptamines are something that people are very interested in because obviously you can have a reduced treatment session. And beyond that, there may be people who are excluded. One of these really interesting areas that you see Dave Olson and Delix and other groups looking at is this idea of psychoplasticians. Obviously, some people will not be people with prior history of sort of schizophrenia or psychotic episodes will probably be excluded from full-blown psychedelic therapy. But for them, it may be possible to get some of the benefits of psychedelics without having that psychedelic experience. So yeah, I think there's a huge amount of clinical optimization to be done here.
Gotcha. One of the other things that you hear from some companies is that most psychedelics produce their hallucinogenic or psychedelic effect through agonizing the 5-HT2A serotonin subtype receptor. But a lot of them, especially psilocybin, also agonize the 5-HT2B serotonin subtype receptor, which some people speculate could cause problems with heart valves. Is that something that you think is a legitimate concern and is also something to pay attention to, I guess?
Yeah, absolutely. If you want to have a look at probably one of the larger data sets that's semi-systematic, Tom Ray published an article in PLOS One about 10 years ago, I think, 2010 maybe, just looking at the off-target binding of a number of different psychedelics at a number of different human receptors and transporters. And what's very obvious from that data set is that these drugs are not very selective single-target drugs. They hit a lot of different receptors. Psilocybin, psilocin specifically, DMT, LSD, they often hit many 5-HT receptor subtypes, dopaminergic receptors, adrenoceptors. So they're what we call dirty drugs in psychiatric drug development. What is known is that 2A activity is necessary but not sufficient for psychedelic activity. So you do
Mhm.
see really interesting compounds like lyseride, which is one stereocenter and one atom removed from LSD, not psychedelic in humans. Very potent at doing other things, is a
Hmm.
2A agonist, but is not psychedelic. And there's a number of these compounds out there. So it's certainly, 2A being necessary but not sufficient. 2B is a serious problem for cardiac valvulopathy. And there have been drugs like fenfluramine that were pulled from the market eventually because of this issue with chronic use. So all
Gotcha.
I'd say on that is the way these things are used historically. Occasional use of psilocybin very unlikely to cause this issue, but for people who are microdosing it at sort of higher doses chronically, that may become a real issue.
Gotcha. So if you're trying to design a new psychedelic drug, you might be thinking we want it shorter acting. We don't want it to hit that 5-HT2B receptor. Maybe we want it to have maybe not no hallucinogenic effect, but maybe a reduced hallucinogenic
Mm,
effect for certain types of people, maybe. Okay. And then there's this question around, like, you said the word dirty drug. And I think this is something that is like maybe not everyone is aware of, is like in pharma, usually people are trying to develop like fairly selective drugs, right? Meaning that they basically have a target receptor and they try to hit that receptor and very little else, right? Whereas a lot of these naturally occurring compounds hit like a ton of different
mm,
receptors. And the question of the effects of those receptors being agonized simultaneously is not, it's not simply just like additive, right? It's kind of like this weird
no.
nonlinear combination. And so it makes it very difficult to disentangle what is causing what.
Mm,
And so on the one hand, you want to start maybe getting rid of some of these like off target, you know, binding profiles, but you also might find strange things like the fact that you can still agonize 5-HT2A but not cause hallucinogenic effects, I guess.
yeah. Yep.
Is there any like leading hypothesis as to what the other thing that is required to have a hallucination is outside
Uh,
of 5-HT2A?
yeah, we know from work out of Brian Roth's lab and a number of his collaborators that beta arrestin is very important. So when the
What is
2A
it?
So the 5-HT2A receptor has a number of different signaling pathways that can be activated when a ligand, an agonist ligand, binds and activates that receptor. It's not as simple as like an on-off switch.
Mm,
It's sort of more like a dimmer switch and like a dimmer switch in a few different colors. So
interesting.
you actually get a whole host of different responses that can be generated in response to a ligand binding. Some of those prefer a signaling pathway that's mediated by G proteins, which is what gives this class of receptors their name. But another important signaling pathway is beta arrestin recruitment. So this is recruitment of a different protein following activation. Most famously, I guess, in things like the opioid receptor, if these are activated excessively, that beta arrestin recruitment becomes sort of preferred, and it allows the homeostasis for the cell to be maintained by pulling those receptors internally to recycle or degrade them. So it's a way for sort of maintaining homeostasis of a system by controlling, how much receptor is available if there's an excess of drug. So we don't think about it.
Hmm.
It's been explored by a number of different labs. Ligand bias is one of the terms, or signaling bias. It's been explored pretty extensively for the mu opioid receptor, a little bit for the cannabinoid type 1 receptor, and to some extent for the 2A receptor. And so Brian Roth's done some really incredible work showing that if you knock out the beta arrestin receptor, for example, the beta arrestin protein, you don't get some of these sort of classic physiological effects in mice
Hmm.
following psychedelic administration. So it seems to be quite a complex, evolving
Yeah.
story.
And I think this is something that, I mean, you kind of said it earlier that it's not a light switch. It's more like a dimmer switch with different colors. And I think that is something that is probably surprising to a lot of people listening because in, the one biology class that someone might take in high school, they sort of they hear about receptor as this basically like a lock and a key, right? But it's really
Yeah.
not that simple. There's many different keys that could activate the lock in some way with very different effects.
Oh,
So,
it blows my mind every day. So the story just gets so complicated so quickly. Very very complex system. So beyond sort of lock and key, you can have things binding to a receptor that actually change the sort of innate ability of that key, to use that metaphor, to interact with the receptor. You can have
hmm,
two receptors coming together and doing the same thing. So one receptor interacting with another one can actually change the efficacy of that of any given ligand for one of those receptors. So it's this really complex dynamic process. And yeah, I think a lot of people don't appreciate that. Talking
yeah.
to people in the psychedelic space, very experienced and knowledgeable people, a lot of them are surprised to learn that there are 2A agonists that are not psychedelic. Like that's still kind of new for a lot of people, which is pretty mind blowing.
Yeah, that's crazy. And given the complexity of like what binding is, this simple like one single scalar value of like binding affinity that you often see, that is like
Mm.
not really describing the full extent of what's happening, right? Because you could have like equivalent binding affinities with different types of things happening within it, right? Is that fair to
Yeah,
say?
for sure. And it gets so binding is typically used in a very simple way. Does ligand X displace a known radio labeled ligand, something that's radioactive that we can measure? And you just look at that sort of equilibrium to see how easily one is displaced by the other. Um, it tells you about how well a drug interacts with the receptor. It doesn't tell you how it activates it. Once you go to functional assays to work out whether something's an agonist, whether it's actually capable of activating the receptor, it gets even more complex again, because typically the assays that we use for looking at psychedelic activity, things like the flipper calcium mobilization assay, there's several steps removed from the actual ligand binding to the receptor and activating it. We're measuring this downstream effect, with one measure. So there's a whole bunch of steps in the middle that are sort of more or less invisible to us with those sorts of systems.
Mm. And interesting. Okay, so let's talk about sort of just the process of taking a starting point, basically saying, all right, we want to create something that's similar to psilocybin, but we want it to be shorter acting or whatever. What is the actual process like? I think a lot of people in the psychedelics world are familiar with Shulgin, and it's like
Mm.
Shulgin basically did all of this work like on a piece of paper and pen. And somehow he was able to invent really cool things like 2CB with that method, right? But now we have all of these computational chemistry techniques and machine learning assisted drug design and high throughput screening. So
Mm.
for a company like Psylo, which you are, now working on, like what is sort of the process like? Like what is step one in this?
Yeah. That's a great question. We've used two different strategies. We've used both an iterative design strategy that's somewhat related to what Shulgin was doing, and we've used computational approaches. So yeah, actually, I have a great slide that I can send to you that I've used in undergrad lectures for sort of showing Shulgin's thinking process. And he started out with like a really simple question. So he's thinking about mescaline. It's a simple phenethylamine, 3, 4, 5 trimethoxy group. So these three different groups in three positions right next to each other around a ring, and he just started asking like how important is each of those and what happens if you move those methoxy groups around. You pretty quickly get to 2,4,5 trimethoxy analog of mescaline, which is still active, changes its sort of potency and duration. You add an alpha methyl group onto the end to block metabolism. You get TMA2. It's even more potent, even longer lived. You start tying some of those methoxy groups back onto each other. You substitute the 4 position for a bromine, and suddenly you're at something like DOB, where the potency is a milligram dose in a human, lasts for 30 hours. And so it's
Jesus.
sort of this, yeah. The DO series are interesting compounds, academically interesting for sure.
Mhm.
So with a few key questions and these iterative cycles of synthesis and sort of testing in some sense, you can start to understand which aspects of the structure inform the activity. So we've done that somewhat, starting with the known tryptamines and making, using really innovative medicinal chemistry techniques to make very specific modifications that we think will retain some of the sort of three-dimensional electrostatic structure of these molecules, but potentially remove some of the liability at 2B and at other targets. So we've done a little bit of work like that. And then on the other front, we've used computational chemistry as a discovery tool. So very large scale virtual screening. So that process, yeah, that's a pretty interesting one. If you're happy for me to jump into it for a few minutes and
Yeah,
describe that
no,
one.
I think a lot of people are not really familiar with
Mm.
that concept. So yeah, what is high throughput screening or just screening in general?
Right. Yeah. So high throughput screening. I mean, we take a step back. Most of the drugs that we've developed they're small molecules. We also have peptides and antibodies and things. But let's just focus on small molecule drugs, things that are like the psychedelics that we know. Uh, Most of these drugs interacting with sort of receptors, enzymes, transporters, other things, a few key classes of proteins. High throughput screening historically was the idea that you would take maybe a proprietary library of small molecules with a bit of chemical diversity. Every big pharma company would have had several different libraries that were, closely guarded sort of um, company secrets. You take several hundred thousand or a few million molecules in these large robotic systems. You have an assay, a test, a screening test that's also set up robotically in large format. And you literally take high concentrations of each of these, tiny micro aliquots of drug, and you test them in this tiny little well to see if there's an effect. So from that, from a million drugs, you might get 5 to 10% of them giving you any sort of activity at a high concentration. Each of those substances then becomes the starting point for a full development cycle. So
Hmm.
lead optimization. So they're often not very potent to begin with. They might have off-target activity. They've got other development liabilities. So you take that chemical matter, you based on a number of decisions, and you start optimizing it on the way to a clinical candidate that's suitable for dosing in humans. What we can do now with all the computational power that wasn't available, even 30 years ago, is to do some of this virtually. So the idea here is that you can take molecules, both real and imagined, you can convert them to a digital string that's representative of the molecule, and then you can essentially look at a number of different virtual three-dimensional conformations of these molecules that are sensible in energy terms, and you have a three-dimensional model of your receptor of interest, and you can literally just sort of, like a baseball into a glove, just throw these molecules at the receptor and see how well they bind.
Mm.
The benefit of doing it computationally is you don't need a physical chemical library of hundreds of thousands or millions of molecules. You don't need a robotic assay system. You can literally do it in consumer grade hardware these days with a home PC.
Right. So this may be a stupid question, but like who comes up with this library of candidates to screen? Is this just something that people can download from the internet? Are these proprietary libraries that you can get?
Yeah, there are both proprietary libraries and sort of public open source libraries. One of the
Mhm.
largest public libraries is run by Brian Choquette and John Owen at UCSF. It's called the Zinc database. They update it constantly. I haven't checked how many molecules are in there in the latest iteration. Let me just have a quick look here. But this thing's incredible. So you can filter these molecules based on their digital representation by physical size, lipophilicity, solubility, all sorts of parameters that are really important for going from, the tens of millions of molecules that are in this virtual library down to a sensible screening set.
Mhm.
Just trying to find the latest version here online.
Yeah,
So they're
I
up
would
to,
imagine that there are hundreds of millions, if not
yeah, yeah.
more.
Zinc
And
12 was 35 million, and they're up
oh
to
wow,
Zinc 20 now, and it's exponential. So I'm going to guess a couple of 100 million or billion molecules in
Jesus.
there.
And are these all molecules that have been not necessary? Some of them are just theoretical, right? They haven't been synthesized before,
Yeah.
but they've been created or ideated by humans, or are these like sort of systematically generated molecules that a computer kind of came up with? I guess
Yeah.
probably both.
Yeah, a little bit of both. So in the Zinc database, you'll find, and there are a number of filters that you can apply to separate these things out, but you can find both approved drugs, you can find drugs that have been annotated in various academic or patent literature, and then you've got this whole much larger class of drugs that don't, as you said, I've used an elegant term there, I can't recall what it was, but drugs that don't really exist, drugs that are sort of theorized to exist or
Hmm.
could be made. Often these are referred to as sort of synthesis on demand molecules. And the
Mm.
reason, they make a number of assumptions. So one of the key reactions in medicinal chemistry and drug discovery is just amide bond formation. You
So
take a
what is
carboxyl,
that again?
a, I think to the US audience, it'll be amide bonds. So you take an amine, you take a carboxylic acid, and you click the two together, sort of like Lego, and you get an amide. We have millions and millions of amines, we have millions and millions of acids, so
Mm.
theoretically every possible combination of these two different categories could come together to give you a new molecule. And that's
Gotcha.
sort of the approach that's applied here. You can computationally generate these very large libraries on assumed chemistry, and
I
sure, there
see.
might be, every now and again there'll be specific cases that cannot be synthesized, but the vast majority of those molecules probably can be made, so they get entered into the database.
Gotcha. So you get your starting library, you probably filter it down based on some criteria, and then
Mhm.
you have your model of the receptor, or I guess receptors in this case, right? It's probably not just 5-HT2A, but, and that's another thing that is like easier said than done, right? There's not like a single perfect representation of a
Yeah.
5-HT2A receptor, right? So is
Yep.
that kind of like an edge too, is having like a better model of the receptor than maybe the next guy?
Yeah, the reason you can have, even using broadly similar technologies, and have sort of different answers to the questions you're feeding in, is that, yeah, everyone's probably using a slightly different, finely tuned model of the receptor, and everyone's probably using slightly different, very large libraries. So
Gotcha.
yeah, you've got a number of different parameters there that are going to lead to different answers.
Gotcha. So for those of you sitting at home thinking like, wow, I guess I can just go download zinc and then like I can make some novel psychedelic drugs, like it's maybe not that easy. Like you there's like there's some thought that has to go into it. And once you've completed this screening, let's say you start off with like a couple million and you end up with 30. The idea is not necessarily that one of those 30 as is going to be the final drug, right? That's more just like a starting
No.
point for further refinement. Is that correct?
Yeah, that's right. It's actually slightly more complicated again, because obviously the docking is not perfect. So docking has both false positives and false negatives. Um,
Mm,
so the case that Psylo's run is a pre-filtered set of about 100 million molecules against our own custom 2A receptor. And from that 100 million, more than 100 million, we've identified 1,200 new hits. So those 1,200 bits of chemical matter can be clustered. They're not 1,200, they're 1,200 unique molecules, but they can be grouped according to similarity in various
mhm.
ways. Some of those will turn out not to actually bind to 2A. So we're in the process of doing this now, taking representative examples from each of these clusters and running them through a traditional experimental binding assay to see if they do actually bind.
Right.
So,
So then this is not in the computer. This is in the
no,
lab,
yeah,
right? Okay.
yeah,
So you're
that's
actually
right.
checking it out for real. And is this like we're not talking in mice yet, right? We're talking in just
No,
some other sort of assay. Okay.
yeah, these are typically expressed receptors in a cellular system that are then harvested and used for this radioligand binding assay that I mentioned before.
Gotcha. And so even then it gets even more complicated, right? Because even if something does bind, that doesn't mean that it is a molecule that is maybe stable or crosses the blood-brain barrier. Like
Yeah,
there are all sorts of
absolutely.
other problems that could happen, right? And some of these you can't necessarily predict. Am I right about that? Like you can't necessarily predict all those things at the beginning. You kind of have to
Yeah, we have a
like try
number
it out.
of yeah, you have to try it out for sure. So the fact that they've been able to be synthesized for your binding assay tells you something about the stability, but they're probably not optimized. So that you're starting again. That compound will be considered a hit. That compound or class. You still need to do this lead optimization cascade. So pluck a
Right.
few atoms off here, put something else on, change this aromatic ring to something else. Try and make it something that has a suitable sort of potency at your targets of interest, something with a suitable half life in rodents, something that doesn't interact with other development liabilities like cardiac ion channels and things.
And this lead optimization process, is this done really just through like, I don't know, human logical thinking, or is there also computer assisted components of this as well, or
Yeah,
machine learning assisted?
yeah, a little of both. So lead optimization trajectories are quite variable. It depends on what your drug target is, what drug class you're starting with. There's definitely a huge amount of human intuition, but also the cumulative experience of lots of people doing medicinal chemistry over decades can inform machine learning approaches and other sort of computationally guided approaches too.
Right. Yeah, I think a lot of times, I mean, I have a academic background in machine learning and did a lot of machine learning at the hedge fund I worked at, and it's like
Nice.
the reality of machine learning is so far from what the popular conception of it is, right? I mean, it's like even the best machine learning stuff requires so much like human tweaking and sort of
Yeah.
human kind of bias injected and even just like the architecture of the machine learning model, right? It's not this idea that you just give it to the computer and like the final result pops out, right?
Yeah. Yeah.
And and both finance, which I was doing at the hedge fund, and like biology are these extremely complicated, like nonlinear systems that are, you can't predict with like simple linear models.
Yeah.
So it's like the idea that you're going to be able to get like super accurate predictions from simple machine learning is, maybe there will be
Yeah.
some breakthrough at some point that makes that the case, but it's certainly not the case now. So you still need lots
Yeah.
of like intelligent human sort of first principles thinking, I think, in the mix, which
Yeah.
makes this all the more fun, I guess.
Yep, yep. Yeah, you
um,
raise a good point about the bias. The people building these systems have this huge influence on the sort of direction and power of those machine learning algorithms that's not really appreciated publicly a lot of the time, I think.
yeah, no, even just like the architecture of the model or like the way that you
Mm.
pre-process the data, this is all a human decision that can have like a very large effect on how useful it is or how generalizable the model is.
Yeah, for
Um,
sure.
yeah, it's definitely something that people don't necessarily think about enough. So, okay, so you you've done some tests, and now maybe it's time to put it in rodents. And this is one of the things that I think is so crazy is that you just look to see if the rodent's head is twitching, right? And that's supposed to tell you if
Yeah. Yep. It's
I
it's a
mean,
remarkable assay, that one, the head twitch response. So it's actually very rare that you get a model that is super predictive. But head twitch, it does have some false positives and false negatives, but it is remarkably predictive for human psychedelic activity, even down to sort
yeah.
of oral dosing potency, which is pretty amazing.
Yeah, I would imagine that it's probably pretty accurate at predicting if there's like psychedelic activity, but that doesn't necessarily mean that the rat is having a good trip, right? Like it,
Yeah, absolutely.
and that's one of the things that I kind of fear when I'm, talking to these companies and they say, "Oh yeah, we put our chemical in and we got the good head twitch response." And it's like, well, that says nothing about, that just means that the thing is tripping. I mean, it could be a horrific, trip through the gates of hell. Like and I think this is part of the problem with like psychiatric drugs as opposed to just like drugs for other parts of the body, is that they may work but may cause like extreme discomfort or unpleasantness
Hmm.
psychologically in the patient.
Yeah,
um,
there are a number of drugs. There's some interesting drugs that are not really good psychedelic um, lead structures, I don't think, but things like efavirenz, which is a anti-HIV treatment. It's actually psychedelic. It gives a head twitch response at high doses. It binds to 2A. People in some parts of Africa, I believe, have taken very high doses as a sort of psychoactive substance. But I'm
oh my
told it's like
god.
it's not very fun. Like it's not a
Right.
really fun experience. So it's positive
Right.
in HDR, but it's not necessarily fun to do.
And there are a ton, I think people don't realize this, there are like a ton of things that are even just naturally occurring that can get you high, and a lot of them are legal. And the reason
Yeah.
that like the government has never bothered to make them illegal is because if you do any of these things once, you'll be like, I'm never doing that again. Like that was horrific, right? Like one
Yep.
example would be like datura, which is a plant that you can just like grows everywhere, and it'll send you on like a crazy trip for like 3 days. But it's like really, it's not fun. No one does this recreationally.
No, you don't need
And
to ban it because no one's out there doing it. Exactly.
right, no one's doing it. And if you just get any of these books, like the like the encyclopedias of psychoactive plants, like you'll find tons of totally legal plants that will make you trip, but are just unpleasant. And so
Yeah.
I always wonder when you're talking about these like novel psychedelic drugs, like is the trip actually pleasant? And you never know until you get into humans,
Until you try it. Yeah.
which may take months or if not years, right?
Yep.
So that's another thing that I think is underappreciated, and why I liked your comment on my post so much, because you were like, well, 10 years maybe not, but 20 years from now, we'll
Yeah.
definitely have some very popular novel psychedelic drugs.
Yeah, for
So
sure.
that makes
I mean,
sense.
Shogun's talked about this as well, right? He had his favorites. He's sort of top 10, and the vast majority of those are not naturally occurring psychedelics. And I think it's just that naturally occurring psychedelics are the most available now.
Mm-hmm.
That's not to say they're the most preferred. MDMA is not a psychedelic, but a good example, enormously popular drug, not natural. I think like
Yep.
psilocybin is just what's well known and what's available now. And I think with a little bit of engineering, I think 20 years from now we'll have both better medicines and probably even better recreational psychedelics.
Yeah, but novel recreational psychedelics will be very interesting. It would be interesting to know if, like, when we're talking about these therapeutic psychedelics, all the discussion is around, can we reduce the trip time? Can we reduce the
Mhm.
hallucinogenic effects? But I wonder, at what point do we get to trying to look for maybe like a different type of trip that is like more
Yeah.
pleasurable or enjoyable or something like that? I think it seems like it would be very difficult to do that even with all the technology in the world, right? But
No, I think it can be done. I mean, Shogun's got a few interesting compounds himself. So one of the reasons he really liked 2CB a lot because it had aspects of a traditional psychedelic and aspects of MDMA in terms of just warmth and socialization. So he always said that was one of his favorites. And Shogun, I had lunch with her one day, and as we were taking a photo, instead of cheese, she said 2CB as we were smiling. So she's a big fan. But then
yeah.
there's other interesting compounds in there too. So things like DPT, the dipropyl analog of DMT. Some people seem to like it, but a lot of people find it very unpleasant.
Hmm.
Things like DIPT, the diisopropyl analog of DMT, profoundly auditory hallucinations. It actually changes the pitch of sounds far more than any
I've heard
visual
of that one
effects. Yeah,
before and have kind of been curious to try it, because
yeah,
but it
yeah.
sounds interesting. Like just
The,
tripping auditorily would be
yeah,
quite
these are
wild.
fascinating like tools for neuroscience. And if you can understand, why DIPT does that, but DPT is less pleasant and DMT does something else altogether, as you start building up this huge body of knowledge, you can potentially rationally engineer kind of any psychedelic that you'd like.
Is there something about the sort of like tryptamine class that makes it more that makes it lend itself to this sort of like modification in new chemical entity space versus say like novel ibogaine or novel salvia type drugs?
No, I think there's the possibility to modify all of these things. Salvinorin A, which is the main compound from salvia, is a kappa opioid, synthetically very challenging to make. Probably fewer sites for modification that are tolerated. I think the reason
Mm,
Shulgin was able to do so much with tryptamines and with phenethylamines is because they have so few sites of modification, but you can make a lot of modifications at each of those sites and end up with compounds that are active. So it's sort of it is
mm,
sort of an innate property of these structures. Any bit of chemical matter as a starting point will allow you to make modifications. A lot of those won't lead to activity, but the tryptamines and phenethylamines so far are sort of the other case where most changes are tolerated within reason, and some are highly preferred in terms of activity.
right. Because it depends on sort of like this, not that I know what I'm talking about here, but like the starting structure may lend itself more than maybe some other starting structure might. And
Yeah,
you will
yeah,
get
for sure.
very different results depending on like what that starting structure is. So when you guys are doing this, are you sort of you looking at the classic psilocybin, LSD as starting points and going from there? Is that kind of the way?
Yeah, we're thinking about it in a slightly different way to a lot of other groups. We're not looking to just modify, DMT or psilocin. We're thinking about changes that will give you a molecule that doesn't just look the same as it's drawn, 2D on paper, but will give you the same sort of electrostatic surface in various conformations. So we're using those as starting points, but I'd say we're sort of a step removed from what most other groups have
Mm,
been doing, which is swapping a hydrogen for a deuterium or tying a chain,
yeah.
tying the nitrogen back onto itself by a chain. We're trying to do things that are a little bit further afield than that in terms of innovation.
Yeah, and like for those who don't know, which is me, like what is the 3D electrostatic structure? What is the
Right.
significance of maintaining that?
Yeah. So molecules are ultimately just electron densities. So you have atoms. The nucleus of an atom is bonded to another atom, and around that bond, we think about them as like this ball and stick model, right? That you've probably seen in high school. Once we
Yeah,
move into the quantum world, it's actually nothing really like a ball and stick at all. It's like two atoms, these two nuclei that are these dense dense parts of the atom with a whole bunch of shared electrons between them. And electrons have like a probability function, so it's like a fuzzy cloud of
right.
places where the electron is likely to be. Depending on what the nuclei of those atoms are in a given molecule, you can sort of have regions of higher or lower electron density. So if you take any sort of two-dimensional molecule as drawn, you can put it into you can make a 3D version of it, put it into a certain conformation, so a three-dimensional shape that it's likely to adopt based on its structure and energy. And each of those conformations will have sort of a unique energy profile and electron density associated with that. So it's this sort of fuzzy cloud of
Got
charge
it.
around a molecule is what we're trying to emulate.
Got it. So you're trying to sort of keep that, like, use the term probability function, which makes sense to me. Like, you're trying to sort of like keep similar, like, probabilities of like electron positions in the
Yeah,
entire. Got it. Okay.
in the entire
Interesting.
molecule. Yeah,
And
yeah,
that means that it has a higher likelihood of having the same sort of binding that
yeah.
the original scaffold might. Is that kind of the
Y-
right way to think
Yeah,
about it?
that's exactly
Yeah.
right. So yeah, you might change things like metabolism, but retain a lot of the sort of receptor interactions that you want, because receptors are ultimately just a much larger um, electron surface, right? That
Hmm.
small molecules bind to them because of the interactions between these fields of electrons.
Gotcha. And you mentioned something else in your long response where you were saying that some of these people are just swapping out hydrogen for deuterium, and
Hmm.
this is a process called deuteration, right? And you see a couple of
Yeah,
different psychedelic companies with this term, like deuterated tryptamines. And it's like that
yeah,
was kind of like the first wave of novel psychedelic molecules.
yeah,
Like, can you maybe go into that? Is that like a super common thing that pharma does in order?
yeah,
And
yeah.
like, what is sort of the thinking behind doing that? Is it just an easy kind of win, or what is it?
So yeah, it's a super interesting story. Deuterium, for those who don't know, hydrogen has a mass of one. Deuterium is this non-radioactive isotope of hydrogen, so it has a mass of two, relative mass here. So doesn't sound that important. What it means is that the bond length for deuterium bound to carbon is slightly different than the bond length for hydrogen bound to carbon. And as a result, the body sort of interprets them largely the same biologically, but will sometimes metabolize them in a slightly different way. So this idea of like deuterium exchange, it exploits something called the kinetic isotope effect, which is sort of what I just described in lay terms. Um, and so there's this idea. There was two companies in the US who were really focused on whether deuterium exchange would actually be viewed by the patent office as an innovative strategy. There was Concert Pharmaceuticals and Auspex, and they were looking at a number of different drugs, basically a whole host of pharmaceuticals that already existed, making systematic hydrogen for deuterium exchanges, and then trying to work out which of these would lead to a beneficial clinical profile. So the first drug that was approved was a deuterated form of tetrabenazine. So this is a drug that's used to treat sort of dyskinesias in Parkinson's disease and other things. The deuterated form has a slightly different metabolism, which leads to more accumulation of this active metabolite, and it has a sort of smoother clinical profile, fewer side effects, sort of longer duration of action, all the sorts of things that you want to see. And the ultimate sort of crux was whether that would be viewed as a novel composition by the US Patent and Trademarks Office. And it became approved as the first deuterated drug. People have continued to apply this more generally because it's fairly clear historically, and it may change in future, but it's sort of, there's precedent there that deuteration is viewed, if it's not obvious, and it was in this case, that it may be viewed as sort of novel IP. And I think that's why everyone's pursued this sort of strategy. Whether there's any merit to doing that, it's not like swapping every hydrogen for deuterium is good. You have to be sort of judicious with where you put the deuterium atoms. And even then, it's sometimes makes a drug worse, right? It's not always a beneficial thing. It makes some drugs better and some drugs worse, depending on how much deuterium you put in there and where it's placed.
And is it generally the case that it is going to extend the half-life because it's like a heavier isotope?
No,
or
absolutely
could it also
not.
Shorten it?
It can also shorten the half-life, and this is one of these things. It's not always a positive sort of benefit when you're modifying a drug in this way. You can definitely get good and bad outcomes.
Gotcha. So it's not exactly like a total like BS strategy, but it's also not the most complicated thing either, right? It's kind of like somewhere in the middle, and that's probably why we saw so many psychedelic companies going after this like right out of the gate, because it's maybe a easier attempt at getting some kind of like novel psychedelic molecule approved. And I think that this is another thing that people need to be aware of, is that there's sort of like a spectrum of novelty. Like on the one hand, you might have deuterated tryptamines, and then on the other end, there's things that are like, so deuteration is maybe like the most simple step. What's sort of like the next step? Like what are some other interesting things that can happen?
I refer to these as like the low-hanging fruit solutions, and you see them applied all the time. So taking one enantiomer of a drug, you might know about Spravato, the ketamine therapy that was just approved as a nasal spray. It's S-ketamine. It's one enantiomer of ketamine, because that's a way to generate IP potentially in this formulation.
And what does that mean for the non-chemists?
Oh, right. Sorry. Yeah. So many drugs historically that we use are what's called racemates. The simplest way to think about it is your hands, right? Left and right hand are non-superimposable mirror images. Because of the way drugs were synthesized historically, many drugs are made as these non-superimposable mirror images. All of the proteins in our body are made with amino acids that are of one mirror image form. So two different mirror image drugs will interact with the body and receptors and things in different ways. So typically one form is preferred and one form is less active or inactive. With ketamine, it's historically been used for a very long time as an anesthetic. It's sold as its racemate, this 50-50 mixture. It looks like S-ketamine may be more useful in depression, even though how obvious that is, I can't really say, but someone's decided that you can make this drug Spravato, which is just this form of ketamine that already exists in its pure S enantiomer form, and sell it as a nasal spray.
So it's basically a mirror image of the other ketamine, and that is like, it's a mirror image, so it's a novel compound basically.
It's not even a mirror image. The ketamine is 50-50 S-ketamine and R-ketamine. It's already
they took
literally half of all ketamine.
Okay. Interesting. They just took half the ketamine.
Half of all ketamine is S-ketamine. It's even less creative.
Wow. Okay. Wow.
Yes.
And and the patent office thought this was worth considering a novel invention.
Yeah, it is a novel invention. So Spravato is approved as that formulation, as this S-ketamine form in a particular solution as a nasal spray for administration in depression is a novel invention. Sort of not too far from that, you have the concept of prodrugs. So these are psilocybin is a prodrug of psilocin, which is the active form, right? Many drugs come as prodrugs. A prodrug is an inactive form of the drug that releases the active drug in the body. Some things need to be turned into prodrugs. So heroin is a prodrug of morphine, that helps heroin get into the brain before it's converted to morphine. So that's the utility of that kind of strategy. You also see a bit of that in the psychedelic space where people are just making prodrugs for the sake of it. I mean, usually you're trying to solve like a delivery or a solubility issue or something along those lines. Psilocybin's already pretty soluble and has a pretty high bioavailability. You
Right.
don't really need to improve that with new and amazing prodrugs. It's already a solved problem. And then deuteration is sort of one step further along in that you get a drug that's more or less the same biologically but might have different metabolism.
Gotcha.
If you keep going along that spectrum, you do what Shulgin did and make actual structural changes that give you entirely new chemicals. And then to go one step further than that, it's like, let's find some entirely new psychedelic classes. So that's sort of the large scale virtual screening. Dock a whole bunch of molecules, see if you can find something that's not a tryptamine, not a phenethylamine, not an ergoline, something that's completely new that still has psychedelic properties.
Yeah. And those three that you just mentioned, I mean, I think some people know those, but like tryptamines, that would be obviously like the DMT stuff. And also
Yep.
psilocybin is a tryptamine as well, or psilocin, right? Is
Yep.
a
That's right.
tryptamine.
Yep. Yeah, they're both
And
tryptamines.
then lysergamides, that's like LSD-ish, right?
Yep.
And
LSD
then
and related things. Yep.
what was the last one again? Sorry.
Phenethylamine. So these are like
Phenethylamines.
mescaline, 2CB, things of that class. Yep.
Gotcha.
And
And
three major classes.
are there any other classes, or is that pretty much it?
Well, it depends how you define psychedelic,
Sure.
right? So there
Sure.
are piperazines that give you a head twitch response in mice that have been sold as sort of designer drugs that people have some psychedelic effects from and seem to enjoy. There's efavirenz that I mentioned before. The anti-HIV treatment is sort of definitely psychoactive in some way that may be considered psychedelic. Um, there's a few other things as well. Mefloquine. There's some antimalarials that do these sorts of have sort of
Mhm.
similar profiles in rodents and in are psychoactive in humans, but whether you can call those things psychedelics,
Right.
very very subjective, right?
Yeah. So within the 5-HT2A agonizing psychedelics, there are those three classes. And is
Yeah,
what Psylo, your company, aiming to? Are you aiming to sort of discover a new class or work
yeah.
within the existing classes?
We're looking at a number of different sort of product families from shorter acting psychedelics, which have an obvious commercial need and patient need, all the way through to sort of sub psychedelic compounds that may have rapid antidepressant effects, and then entirely new classes of psychedelics, things that may be antidepressant in nature but not hallucinogenic at all, much like what Delix is doing. And
Mm-hmm.
across each of those product families, we're looking at both iterations of existing psychedelics and then leaning more and more into this sort of large scale computational discovery of entirely new chemical matter.
And this is really what I think you guys are doing that, as far as I can tell, stands largely alone amongst the other psychedelic companies, because most of the other companies are just looking at these more low hanging fruit
Mm.
modifications of the existing psychedelics. So you guys are really, going out with a bold vision of kind of, discovering a new class and creating
Yeah,
this entirely new sort of like experiences, whether experiential or not, but these sort of entirely new classes, which is very cool.
yeah. Oh, thank you.
It's a cool vision.
Yeah, it's. I think it's possible. I believe it really is possible to do that. And this has been informed by sort of work that I'd done in the cannabinoid space, and then working with people from Brian Kabilka's lab at Stanford who are working on new opioid ligands of this type. So, THC is not the only class of psychoactive compound that interacts with CB1. That's how we got into the whole synthetic cannabinoid MPS gray market fiasco in the first place. Many other classes of molecules can also interact with CB1. Mu opioids kind of the same. So we've got morphine, we've got fentanyls, but there's actually dozens of other classes. Some of Brian Kabilka's work with Brian Choquette and others has shown that if you apply these computational approaches, yeah, you can discover an almost infinite number of chemical starting points for new opium-like drugs.
Hmm.
Once you start applying that to psychedelics, the story is a bit more complicated, as we've said, because it's not just a matter of designing a 2A agonist, but along those same lines of thinking, there absolutely has to be completely new classes of psychedelics out there for sure. So it's just a matter of
Yeah.
directing efforts in a sensible way to discovering them.
Yeah, it's like so exciting and interesting, on the one hand, and then on the other hand, you'll have people that, like, I think someone commented on my question on Twitter, basically saying, people put lots of resources into trying to discover new cannabinoids, and like some of them were successful, but like by and large, people just smoke weed if they want to smoke weed,
Yeah.
right? Like no,
Yep.
most people are not going out there for synthetic cannabinoids. And obviously in psychedelics, it's a little different, right? Like a lot of the popular psychedelic drugs are synthetic, but um, it's tough to imagine, coming up with something that is, it's easy to imagine coming up with something comparable, but something that is like preferable
Mm,
is an even taller order, right? And when you're
yeah,
getting
for
drugs
sure.
approved by the FDA, it usually has to be like an improvement on what's already there, right? And so that's kind
Yeah.
of like that's the big challenge.
Yep.
But it sounds like you guys are up for it, so I have high hopes.
Yeah. Yeah. No, we're definitely excited about it. We feel somewhat vindicated. Brian Roth has mentioned this in his latest piece in with Tristan McClure-Bagley in Nature Reviews Drug Discovery. He said everyone's it's the early days of this sort of psychedelics renaissance, right? Everyone's focused on sort of near-term commercial goals and on working with substances that we sort of already know and understand pretty well. But his sort of longer-term view is that, fundamentally, like with other classes of drugs, antipsychotics and other things, the evolution of the field will be into these like much more interesting and rich areas of sort of chemistry and pharmacology, drugs that have quite different profiles, different structures, emulate some aspects of the psychedelic experience, but not others. So yeah, I think it's important to appreciate that we're just very, very early on in this whole sort of
Yeah.
emerging industry, and we're taking a very long term view of the space.
Is there something about psychedelics versus cannabis which you could use to sort of make the argument that drug discovery and these sorts of pharmaceutical techniques will be more successful in psychedelics than they have been in the cannabis space?
Um, I don't know. It's really hard to say. I think the models are quite different. Obviously, cannabis is enormously popular as a sort of recreational intoxicant as well.
Hmm.
Its medical use sort of almost followed recreational use in many ways. With
Yeah.
psychedelics, there's a little bit of that, but I think there is a much larger medical opportunity here. There are far more people who benefit from medical psychedelics than from medicinal cannabis potentially in terms of conditions that are extremely hard to treat currently. So I think it will be quite different to cannabis. Cannabis is already very widely loved by
Yeah.
many people across the globe. THC is a pretty good drug for a number of reasons. In the same way that psilocybin is also very much enjoyed and a perfectly suitable drug for a lot of people, there will be other people for whom it's not suitable. For various
Yeah.
reasons. So I think there's
Yeah.
more scope to be done in the psychedelic space than there is in cannabis.
And even just starting off, going back to kind of the beginning of the conversation where we talked about like what are the problems, quote unquote problems with psilocybin.
Mm.
It lasts like 6 hours. Cannabis doesn't
Yeah,
last 6 hours, right? Like
yeah,
it's like much shorter.
yeah,
It also, it's not nearly as intense as psilocybin is, so you don't really
yeah,
need to worry about like reducing that effect. So it's already maybe more optimized for like clinical use than
yeah,
psilocybin in its natural state is. So maybe there's
yeah.
less to improve on, which is something
Th-
that people might want to think about.
Yeah, for sure. I don't believe there's no perfect drug. There's no good or bad drugs. There's just good and bad uses of drugs, right? So THC in the form of herbal cannabis, it's pretty good. If you want something that's short acting, you can just vaporize or smoke some cannabis. If you
Hmm.
want something that's longer acting, you can make some edibles or an oil or something. There's already so many ways you can work with cannabis itself that are not going to be, you know, directly sort of portable to psilocybin containing mushrooms or other psychedelics.
Yeah. One totally switching subjects. One of the things that I know we wanted to touch on was something that you also talked about in the Hamilton Morris podcast about this company in the Ukraine that
Mm,
is having some issues at the moment because of the war situation.
yeah.
Can you talk a little bit about that and explain what's going on there and how that might affect the psychedelics industry?
Yeah, so it's not a super widely known fact outside, the world of pharmaceuticals and medicinal chemistry, but um, the sort of really creative and clever Lego blocks of the drug discovery world, to use a metaphor, are not universally distributed. These are sort of clustered in various parts of the globe. Although huge amounts of pharmaceutical development goes on in the US, a lot of the building blocks for the discovery stage of pharmaceutical of development is coming out of Eastern Europe. So this company that you're referring to is Enamine. So they're based in Kiev in the Ukraine. Yeah, phenomenal company. We've worked with them. I've worked with them in my academic roles for many years, and even now with Psylo. They provide, they synthesize something like 2,000 new building blocks a month.
Oh.
They've got these enormous catalogs of building blocks. When I was talking about making amides before, coupling an amine with a carboxylic acid, they're the people that make the specific variants of carboxylic acids and amines that you might want to click together. So
Mm,
yeah, hugely important group. After Russia invaded Ukraine, they were disrupted enormously, and that's had flow-on effects to everything from very early stage small biotech startups like Psylo, all the way through to larger pharma companies who have discovery programs in a number of areas. So yeah, super
gotcha.
important company.
And so they're synthesizing a lot of these sort of trial chemicals that you're looking at, and if they're not, and are there other companies elsewhere that have the same capabilities, or is it just not that simple? Like there's a lot of kind of maybe knowledge that they have that other people don't have that
Yeah.
makes you can't just like call up someone else in the US and get the same thing quickly.
No, no, that's right. It's because it's not just if you know what sort of building block you want to make, sure, any chemist in the world in theory can make it, but there's a creative element that comes from a huge amount of experience in that part of the world, and the
Mhm.
history of these companies. So my understanding is a lot of the Eastern European chemical companies formed at the fall of the Soviet Union, and it's pretty interesting that the sort of Soviet pharmacopoeia is very different to what exists in the Western world. They've got drugs approved there for use that you'll never see approved in the US or Europe. And the same is true of Japan, actually. There's sort of been several different parallel pharmacopoeias developed through human history. Once the Soviet Union collapsed, a lot of those companies became building block suppliers. And yeah, huge amount of creative chemistry going on that can't easily be replicated elsewhere.
Wow. Yeah, I've heard people that, know a lot more about this than I do talk about just a lot of these old Soviet drugs, like a lot of the nootropics that is kind of
Yeah,
sort
right.
of similar to the psychedelics, but a lot of that stuff came from Russia. Is that right?
Yeah, that's right. Yeah, yeah.
Yeah, it's
They've got a lot
interesting.
of wacky stuff there. They've got like I'm trying to think of a good example here. I mean, I think a CB1 antibody, so something like an antibody that blocks the cannabinoid receptor, was marketed as a drug there as like an
Wow.
appetite suppressant and stuff. Things that
Huh.
you just would never see approved in the US.
Super interesting. Yeah. So I mean, do you have any idea what the status of that is? I mean, are they on hold? Has their entire production facility been destroyed? Like, is do you
No.
do we know?
Oh, the emails. I mean, they're really just really impressive people. Extremely resilient, and it was pretty remarkable getting emails from people at Enamine announcements during the peak of the invasion about how they've had to suspend operations for a few days because they're being actively shelled, which is just
Jesus
like
Christ.
it's just wild, to think that people are
Yeah.
still like going to do their jobs because they
Yeah.
yeah, I mean it's crazy, but as I understand it, they're more or less fully operational again now.
Oh, that's good.
But they were disrupted for a little while there. But this is Ukraine is still under Russian attack. It's still going to be a problem sort of longer term. So
Jeez.
yeah, I think Brian Choquette and others who have built the zinc database using information from a lot of these chemical vendors, they had a commentary very recently about just it's extremely important that the world starts paying attention and makes sure that these people are safe and that there's sort of financial aid there for because it impacts pharmaceutical development in every part of the world quite literally.
Yeah. I think that's another thing that a lot of people don't realize is that I think when people think about these biotech pharma startups, they kind of imagine that just everything is done by like the scientists that work at those companies. But the amount of outsourcing that happens is like
Yeah,
incredible, right? Like
yeah.
even the clinical trials themselves are often outsourced to some CRO or something that does them, right?
Yeah, this is
Um,
the pharmaceutical industry at every scale is completely subject to globalization in the same way that almost all industries are these days. So it's often not, Psylo's doing a lot of our chemistry in house now because we've got a lab set up and built. But before we did that, we were partnering with contract research organizations in China and the US to do synthesis for us. We're still doing some of our receptor screening in China. We'll still do some of our animal experiments in Taiwan and in the US with NIDA. So yeah, a lot of what we do is still quite decentralized and contracted out to different groups around the world. Yeah, clinical trials are exactly the same. Australia is actually one of the more desirable locations for conducting clinical trials because we have this very generous 43.5% tax credit for R&D. So a lot of big
mm,
pharma companies will actually do their clinical trials here in Australia. We've got a whole infrastructure built around that.
wow, that's very cool. Yeah, having a big R&D tax credit definitely a benefit. So look, Sam, we've done almost an hour. I don't want to take up too much of your time, but like what's sort of on the near term roadmap for Psylo? Like what should people kind of, keep an eye out for?
Yeah, it's a great question. I mean, we're hugely excited. We're a relatively new company, but we've managed to do quite a lot. We've generated a library now of about 160 compounds that are iteratively designed. We've got more than 1,000 now of these computationally discovered molecules that we're validating and optimizing. So we've got a huge amount of data around our existing pipeline. We have sent our first set of compounds to some collaborators at NIDA to see if these will induce head twitch response. And we've also set up a partnership here with a government science org to look at the ability of our compounds to induce neuroplasticity in various ways. So yeah, we're just hitting a super exciting stage with Psylo. And I'd say check out our socials. Psylo.bio is the website, and pretty active on Twitter and LinkedIn and things as well.
Awesome. Yeah. And we can't, just because you follow him on LinkedIn or Twitter doesn't mean that he's going to send you samples. Okay. This, you have to wait for the clinical trials to get approved.
Yeah, I should be clear. We're not Shogun. We're not. It's pretty amazing in this space. You will occasionally hear rumors of various executives trialing substances from their own pipelines, all sorts of crazy things. I assume that. I'm going to assume they're all rumors, but yeah, I could say we're definitely not one of those groups.
Of course not. No. That makes sense. Awesome, Sam. Well, hey, thank you so much for taking the time to talk. I really appreciate it. Did you already give your Twitter handle? Where can people find you specifically online?
Yeah, my Twitter handle. I can't actually remember off the top of my head. I think it's at Samuel_B_PhD.
There we go.
So check me out on Twitter.
PhD. Good alliteration there. I dig it.
Pretty active on Twitter. Mostly dissing the entourage effect in various forms, as you're
That's
aware,
right.
Bro.
Yeah. If you want to fight about the entourage effect, follow Sam and tweet at him. All right. Are there any other closing thoughts you have, Sam, or you think that's a good place to leave it?
No, I think that's a good conversation, and yeah, thanks so much for having me on, Brom. I like speaking to people on on the sort of VC side who are just super into this space and really across the science and really interested in sort of a science-driven approach to investment. So appreciate you taking the time.
No, thanks so much, Sam. Appreciate it, and look forward to doing this again sometime. Cheers.